I'm going to be talking today about so-called new approach methodologies in the assessment of immunogenicity during drug development. And I'm going to give you a relatively broad overview with a few specific examples. In many of these instances, you can follow up with the references that are given on the slides themselves. So let's start with a very brief introduction about immunogenicity and NAMs or new assay methodologies. So first and foremost, immunogenicity. When we develop a drug, why do we care about immunogenicity? And I like to think of immunogenicity as a sort of invisible wall, which has immense costs to the patient. And here I've illustrated, the across different protein therapeutics, the economic gap between somebody who responds to a drug and somebody who does not respond to the drug due to immunogenicity. So we are talking about extremely expensive drugs, which can cost in the range of $25,000 to $300,000 a year. And for a non-responder, that cost can rise very easily to over $1 million. So there is a obvious large increase in the actual cost of the drug itself. When a drug stops working because of immunogenicity, there is increased hospitalization, and that's essentially a resource drain. And there is greater utilization of emergency rooms in the US. In other countries, there's similar kind of other avenues that are visited, and that extra managed care results in crisis spending for the patient or for the healthcare system. And then there is loss of productivity. Many of the people on these drugs anywhere lose several days a year from work because of the underlying disease, and if you don't respond to the drug, that only goes up. So it's an indirect economic hit to the person who has the disease as well as to the society as a whole. So what immunogenicity does in essence, it turns high cost biologics into very expensive tap water. And so it behooves us not only to tackle immunogenicity in the clinic, but to the extent possible, develop drugs which have as low immunogenicity or are immunogenic in as few people as possible. To move to NAMs, that's a relatively recent terminology for methods that have been actually used for a while. And a few years ago, FDA laid out its roadmap for reducing animal testing. And the legislative foundation for this was a 2022 act that formally legalized the use of non-animal methods in new INDs. And importantly, it removed the 1938 statutory mandate for animal testing. And the scientific rationale for this is that 90% of drugs or more that are safe or effective in animals fail in humans. And animal models poorly predict human response, and that is especially true in immunogenicity. And any one of us who has done any biology knows that when we want to raise antibodies or we want to create reagents, we inject a protein into an animal. So animals are prone to react very strongly to human proteins, and you're not going to get a lot of information from doing these kind of preclinical studies. So there is not a guidance yet, but a draft guidance for general considerations for the use of new approach methodologies in drug development which can be downloaded from the FDA website. And NAMS is an umbrella term that includes a broad range of methods such as complex in vitro, 2D in vitro, in chemical, and in silico studies. And we will, go through some of these specific studies, specific methods as they apply to immunogenicity risk assessment. So when it comes to NAMs for immunogenicity assessment, there are in silico approaches such as T cell epitope predictions, the net MHC2 or net MHC1 pan being the most popular. There is artificial intelligence and machine learning based structural modeling of the immune complexes. There are population-based HLA diversity analyses. In vitro methodologies include the maps or the MHC-associated peptide proteomics that are mass spectrometry-based assays to identify epitopes. There are peptide HLA binding assays. There are dendritic cell maturation and activation assays. In ex vivo platforms involve HLA-stratified PBMCs in T cell profiling, cytokine profiling, the whole blood assays from diverse donor cohorts. And the list is quite extensive. And the last 2 or 3 decades, there's been tremendous progress in using these assays, both in academia, but more importantly, by industry during the early stages of drug development. So let's think of the immunogenicity cascade and as a series of events that are, and this is a rather simplified listing of a very complex process. But the reason I've put it in this simplified manner is that each one of these particular points actually allows us to develop a particular set of assays to interrogate different steps in the immune response to a therapeutic protein or any protein that is used in a therapeutic application. So first and foremost, there is the internalization of the drug by antigen-presenting cells. There is the processing of that protein into peptides. There is the presentation of these peptides on either HLA class 1 or class 2, depending on how the peptide was generated. And it was generated extracellularly and fed to the cells, or happened to be generated intracellularly itself. For example, if you inserted an mRNA and got it to make a Cas protein, for example, that would be an example of something that would take on the HLA class 1 pathway rather than the class 2 pathway. Then there is the recognition by a T cell receptor and activation of either CD4 plus T cells or CD8 T cells, resulting in their proliferation, and finally the development of antibodies. Now, most NAMs measure one step in this complex cascade. And there is clearly an unmet need to develop fit for purpose workflows for individual applications. And something that's just beginning to emerge now, but will, in my opinion, develop very rapidly, is the use of AI tools and mathematical modeling, which will provide new opportunities for better interpretation of data obtained from diverse different NAMs, which can together be more clinically relevant than it is today. Now, when we talk about immunogenicity, there is, one huge elephant in the room that we always need to address, and that is the human leukocyte antigen diversity, or the HLA, or the MHC diversity. I mean, HLA is the same as MHC in humans. So the HLA genes are the most polymorphic genes in the human genome. And when you get blood bank donors, you have a selection bias. And in addition, because of the sheer diversity, the human population has thousands of these HLAs. Even a random sampling, unless it's phenomenally large, will not give you a representative population of a donor cohort. And one solution that we have used something called simulated annealing to develop an algorithm that optimizes a cohort to match the target population for HLA distribution. And by target population, I mean, if you are interested in the world population, you can get the world population as your target population, but there are large differences between populations in terms of the relative composition of the different HLA alleles. So for example, a Japanese population may not have the same distribution of HLA alleles as a US population or a North American population or a population from sub-Saharan Africa and so on and so forth. So what this algorithms that use computational tricks like simulated annealing do is it allows you to make these kind of selected populations. And this graph over here on the x-axis is something called a Janssen-Shannon distance score. And you don't have to understand the statistics of it, except that the larger that this score is, the further you are away from the desired population. And what this graph over here shows you is that you have 10 million randomly selected donors, and they are still quite far apart from the desired population. On the other hand, if you select just 50 donors using this algorithm, you can get get an almost 7 standard deviation better selection than using random selections alone. Now, to move on and to give you, some examples, from case studies, from our publications, from other publications, etc., of a slew of NAMs that span from the in silico to complex in vitro and ex vivo tools. The first, the simplest, and the one that has the highest throughput are in silico approaches. So essentially, most of the existing in silico tools, what they tell you is how well a peptide of interest will bind to either HLA class 1 or class 2 molecules. It's an early initial way of recognizing if some epitopes in your protein are likely to be immunogenic or not. But remember, even at its best, even when this algorithm works very, very well, this algorithm can only tell you whether the proteins developed from the peptides that come from your protein, do they bind HLA or they do not bind HLA? They don't really tell you whether you're going to get an immune response because of this binding. It's an early and necessary step in the immune cascade, but it's not a sufficient step to get an immune response. So, you can get these scores and you can use them to generate a promiscuity to score to give you a sense of what the population risk is likely to be in your population of interest. And several validation studies have been carried out, and these tools are now, especially NetMHC, are very, very accurate in the sense that in terms of specificity, that is correctly identifying non-binders, they're about 98% accurate. They have good sensitivity, identify naturally presented MHC ligands with 90% accuracy, and they have a AUC of about 0.95 in discriminating between binders and non-binders. And here I've given an example of vetropitok alpha, a drug that was being developed and was found to be highly immunogenic and remote from development, because there were, you can see in red these sites that were inserted into the wild type protein, and they created these neo epitopes. So you can see in terms of peptide affinity, these new epitopes that were created are really lighting up over here when you take many of these donors, which are on the y-axis. And here is a promiscuity score, and it's their promiscuity scores for different populations. And you can see that for some populations, the promiscuity scores are slightly lower. For others, they're very high. So what a promiscuity score does is that given the HLA distribution in that population, it will tell you how likely is that drug likely to be immunogenic in that particular population. Here's an example of another NAM, which is increasingly being used, a very sophisticated tool called MHC-associated peptide proteomics. And it's the only method that can measure both peptide processing and presentation. It identifies peptides bound to both HLA class 1 and 2 molecules. Peptides identified show high affinity for donor HLA. Here you can see that, in one example, for one donor, if you look at the in silico assessment of peptide affinity, the peptides bind with very, very high affinity based on this percentile rank. If you take 1 million random peptides or peptides from a large protein like factor VIII or 1 Wildbrand factor, they are all over the place. Like they have the same, essentially the same probability density. Whereas peptides that are actually bound have very, very high affinities. Peptides identified correlate with T cell activation in vivo assays. And this method avoids overprediction, as only peptides actually presented by the antigen presenting cells are detected. Here's a MAPScape case study where this is the same example I gave you earlier of the highly immunogenic variant of factor VII that was generated and found to be highly immunogenic. So this is a cluster frequency is a way of measuring how many of the peptides are bound to the HLA of interest. And compared to wild type, you can see that you had many more, you had a higher cluster frequency for this drug, vetropitok alpha. And when you make mutations to deliberately decrease peptide MHC binding, the cluster frequency also falls. And, this is the corresponding T cell responses. You had 0% for wild type, 12% for vetropitok alpha, and it goes down for these deimmunized variants. And as far as clinical anti-drug antibodies are concerned, this wild type factor 7A used for more than 20 years. There are no reports of immunogenicity. 11% of people in the clinical trial with vetropitok alpha got immune responses, and these have never been used in clinical trials. This is an example of T cell activation assays, and increasingly we have novel modalities like Cas proteins, vectors like vectors for gene therapy, etc. Here, this is a study by Charles Worth et al. Looking at pre-existing immunity of Cas9, SA and SP Cas9 proteins. And again, you are looking at HLA type donors and using PBMCs for, you can use them for, multiple readouts. And in this case, they're looking for percentage positive cytokine producing T cells, and compared to unstimulated cells, cells stimulated with SA and SP-Cas9 are almost comparable to what you have if you challenge these same cells with a tetanus toxoid. Like I said earlier, individual NAMs give you limited information. This is again, the same case study with factor 7A and its variant, and looking at different assay methodologies. And you can see that, in a post hoc assessment, these are in silico and vitro methods. This is the MAPS method. And this is asking the question of, do mutant peptides that bind with higher affinity elicit a T cell response? And the answer over here is also yes. And all of these have an association with clinical immunogenicity. In addition to in vitro and ex vivo assays, there is increasing use of machine learning tools, not only to integrate data got from NAMs, but also looking at clinical data. And in this example, 6 different machine learning classifiers were used to select the best machine learning classifier, and then use this so-called mean SHAP value to rank different patient attributes in terms of what elicits the largest immune response. And here you can see that genetic factors such as factor VIII variants and HLA repertoire dominate as the predominant factors that are good predictors for immunogenicity to the factor VIII protein, which is used to treat hemophilia A. Interestingly, race, which is anecdotally associated with clinical incidences of immunogenicity, ranks pretty low. And that is because, in my view, many of the specific attributes of race are already incorporated into these genetic factors, which are actually better predictors than race per se. Now, so unbiased variable identification can be done using machine learning, and there are HLA associations across therapeutics. And these are some publications which have carried out very large studies for TNF alpha inhibitors. The other publications on factor VIII treated to treat hemophilia, etc. And many of these studies show that there are HLA variants that could put a patient at either high risk or actually protect them from getting immune responses. And because HLA the composition of HLA alleles in different populations varies so drastically, it is, again, in my personal view, important that global trials should consider HLA diversity, because, some of these, if you pick a population where some of these HLAs occur at very, very low frequencies, you're likely to miss some of the risks associated with a particular protein. This is an example of using multiple NAMs to circumvent immunogenicity, where many of the techniques I talked about just now are used and integrated to redesign this molecule, the vitropatog alpha, which was very immunogenic, to, to make variants of that molecule which retained the functional characteristics for which the design was made initially. But this graph shows you very clearly that the vitropatog alpha compared to the wild type protein was much more immunogenic in terms of stimulation index in an in vitro assay, and the variants designed are much less immunogenic. And again, this entire workflow has essentially been designed using a variety of NAMs, and it completely bypasses any kind of animal study. I would like to switch gears and move on to novel modalities, which is gene therapies, mRNA, LNP, cell therapies, and gene editors. All of these, for all of these, there is an unmet need for new workflows and new methods, essentially, for predicting the immunogenicity of these molecules. And one of the major issues for many of these modalities is that, unlike therapeutic proteins, these are synthesized in vivo. And therefore, for example, if you were to do a MAPS assay, and here is an example of a new workflow that we developed to identify class 1 peptides that bind to class 1 proteins. But not only do they bind to class 1 proteins, but the whole workflow ensures that the protein is actually synthesized in the antigen-presenting cell itself. And here is, the list of, peptides that were identified on a Cas9 protein when antigen-presenting cells themselves produced the Cas9 proteins intracellularly. Here is a sampling of emerging NAM technologies, which we are not using as much today as we should be. So there is the ability to do TCR repertoire sequencing. There is ability to do paired antibody sequencing and very high throughput peptidome profiling. And the widespread use of these techniques could generate data sets for predictive algorithm development and not only improve mechanistic understanding of immunogenicity, but create personalized risk assessments for patients when these are brought to scale, and the cost can be brought down to a reasonable amount that can be used routinely in drug development and in the clinic. There is another I would like to provide as a sampling of new reagents that have been used in the last 3 or 4 years. And one very powerful tool are the so-called ankyrin repeat proteins, which can be constructed with closely packed repetitions of amino acid residues. And Ankyrons are a commercially available variant of these proteins. And we use these Ankyrons, which are promising. They essentially perform the same function in analytics as antibodies, but these are artificially generated. You don't have to resort to animals. Again, the essential theme of this talk. And I just wanted to give you an example of, Ankyrons that were designed to bind with high affinity to the SARS-CoV-2 virus for different variants. And what was done in this study was we looked to find Ankyrons which were raised against one variant of the SARS-CoV-2 and that could cross-react with other variants. And what you see over here is that these Ankyrons, there are several of these Ankyrons which have broad neutralizing power to neutralize very diverse emerging viral strains. So as I approach the end of the talk, I'd like to spend some time on unmet needs and collaborative opportunities. The first of them is, of course, the need for standardization, a dying need for reference reagents and quality standards for NAM reagents, for ADA and NAB assays, harmonized protocols across laboratories, validated cut points and acceptance criteria, because we make decisions about is this drug immunogenic? Is this drug immunogenic in this individual, or is it not immunogenic? And many of these decisions are based on generating a threshold called a cut point, and it is important that these are done in a very thoughtful way. There is the need for data integration, and I told you that previously that this is an emerging application for AI and machine learning, which will become very, very powerful, because individually we make judgment calls based on getting data from different NAMs. These tools can offer a more unbiased approach towards consolidating data from different studies. And then there is, of course, the need for collaborative platforms between industry and academia, and more public-private partnerships, and eventually to use international bodies like the ICH to develop guidelines and guidances which can be adhered to by people all over the world. The takeaways is that we have a good foundation of multiple validated NAM tools, and there is some limited proof of principle for concordance with clinical outcomes if the experiments, donor pools, etc., are carefully designed, as are the workflows. Most importantly, NAMs are human relevant and can and have provided mechanistic insights. The pathway for implementation of NAMs is the FDA Modernization Act 2.0, which enables NAM acceptance in a regulatory framework. There is a roadmap for implementation which has been provided, as is a guidance document. The practical benefits are reduced animal use, which has both an ethical and a scientific imperative, lower costs, faster development timelines, and better prediction of human responses. And the future vision is of an integrated multimodal assessment that includes in silico, in vitro, and ex vivo, and population personalized level risk assessment. And most importantly, as we come into an era of different modalities, not just therapeutic proteins, these NAMs offer a basis for generating workflows where you can pick and choose different NAMs that are specific to the questions related to the drug or the medication or the approach that you are developing. I'd like to thank the people. I'm very grateful to people in my lab whose photographs that you can see here and read their names. My collaborators for the clinical data, the so-called My Life, Our Future Consortium, where hemophilia patients donate their genetic sequence and also information about their incidence of anti-drug antibodies, etc. I've collaborated with several members of the industry. Data shown here includes data from Editas Medicine, CSL Behring, Novo Nordisk, Abzena, ProImmune, and ImmuneSpec, and to all of these, I'm very grateful. And finally, these are the sources of my funding. Because I'm at the FDA, we don't take funding from outside, so they're mostly all internal funding sources. And I'm happy to answer questions, but if you have a scientific question at any later date, you're always welcome to email me at this address with the caveat that I am happy to respond to scientific questions and scientific issues. Regulatory questions, of course, you'll have to follow the regulatory pathway and not email me directly. Thank you very much for your time, and I hope this has been useful to you.
Thank you very much indeed, Zuben. That was a fantastic talk. So I'm going to pass on to the audience that if you all have any questions for Zuben, we'd like you to you can raise up your hand, let me know. And then because we'll be using the laptop's microphone, so we need you to move up here to ask the question. Yeah, that's okay. Yeah. So we have a question for you, Zuben. So I'm just inviting the person up to the laptop. Yeah, we can actually go there and turn on the video. Yeah, I think it might be good. Turn on the video, at least you can see Zuben. Zuben can see you. Can see us okay, Zuben?
Yeah, perfectly. Hi.
Can you see? Hi, nice to see you. And it was great to talk. And I was personally very interested in like artificial intelligence model for predicting some announced models. And honestly, you mentioned that as one of the weakest point of the in silico nums is like a standardization. So, but you listed some methods in silico method as the net image pan and as the in vitro as maps. So is it regarded as, although there's no standards, but is it just you brought up those examples because people widely use those tools, or you FDA or you personally take those tools as a standard at the moment?
So you are right. There are I think the net MHC among the ones that are non-proprietary and in the public domain, the net MHC has gone through a considerable amount of validation and standardization. But again, that's largely done by the group that developed that particular algorithm. There is no industry-wide, or there is no central academic attempt to independently validate many of these methods. And we with the net MHC, we have reached a point where I think there is a general consensus that the method is very, very accurate. However, like I mentioned earlier, it is accurate in doing what it is supposed to do. It will tell you whether or not a peptide of interest is likely to bind to your HLA of interest. It's not really going to tell you much more than that. The next step is something that you are just presuming that if it binds with very high affinity, that information is useful with respect to immunogenicity. So machine learning has been used to develop this technique. The other machine learning tools that I'm talking about, which are still in their nascent stages, or artificial intelligence, is, right now, let us say you start with your in silico, then you go up, do some in vitro, you get, a whole bunch of data with, some in vitro, some in silico, some ex vivo studies. And then you apply your judgment, and judgment comes with bias. And you take this entire data set and make some conclusion about the immunogenicity risk of your molecule. And I think there is a room and a place and uh, a good chance for AI and machine learning to consolidate all these different data sets and come up with a meaningful, clinically relevant outcome that goes beyond just whether a peptide will bind to MHC or not. Something that is, much more holistic than that.
Sorry, I have another question. So I have some collaborators who's working on organoids to predict the immunogenicity, and there's some special chips that contain like the PBMC like stuff in the chip, and to mimic the human immune system. So are FDA open to those kind of like new technologies to predict the immunogenicity, or you tend to wait for like the proven record of those technologies?
So, at this point, I don't know what you mean by is FDA open to these technologies. Any data that you bring, we are open to, because, it's basically, a science-based decision on whether, what information you bring about your drug and its immunogenicity. Does it make sense, or doesn't it make sense? At the end of the day, however, in most instances to date, we make our judgments based on, the assays as they are carried out on clinical samples and clinical studies. So the larger value of many of these early studies is for the company itself to develop a better product that will either be less, you obviously don't want one that is more, unless you're making a vaccine, you don't want a product that is more immunogenic in the clinic. So that's, so in terms of, in terms of openness, yes, we are open to everything. We don't shut our door on anything.
Thank you, Zuben. So we have another question. So I pass on the mic to next person.
Thank you. Hi, Zuben, can you hear me? Thank you very much for a nice talk.
Yeah, thank you. Of course, I can hear you, yes.
Okay. Hi, this is Zuben from GenScript. I actually use work on gene editing methods, and actually I'm really interested in the topic that you mentioned for example, for Cas9, for gene therapy using Cas9. The Cas9 protein actually contains peptide, could cause immunogenicity, and the solutions of it. Actually, I'm wondering, like when you do the prediction and in silico model, would you also considering combines the delivery methods? For example, for this Cas9 or gene editing tools, we can deliver the virus to human that's actually quite long-term consistent expression of the Cas9 protein that definitely will cause a severe immunogenicity. However, on the other side, if we use like non-viral methods like mRNA, the expression is transient. Once it's become protein, actually go to the nucleus directly, and then they stop the editing, and then it's done. Actually, I think it actually can prevent the immunogenicity issue. Would like the model to predict the impact, we also combines how the format of this tool and how it's being delivered to the human body. And also, in the end, we could give even better suggestions on what type of disease modality, what delivery methods we should use for treatment.
Yes, so you asked a very good question, and that goes to the heart of the issue with novel modalities is that, we have with protein therapeutics, you have, many different proteins, but for the most part, they're all delivered in more or less the same way. You make a highly purified protein and you inject it into a human being. Some may be, there may be some, differences about, locations where you inject them, or, whether they are intravenous or intramuscular or whatever. With novel modalities, or even with the same modality, there are, depending on the company, depending on what your approach is for the disease, you're going to deliver it in very different ways. And that's where, the immunogenicity assessments for these modalities will have to be much more thoughtful than, we've had previously for protein therapeutics. And the reason, I gave you one example of, we have a publication which looked at, the identifying epitopes on Cas9 protein, which when it is delivered as a protein via the class 2 mechanism. Since then, and we haven't yet published it. It's under review right now. So we developed actually developed a whole different workflow where we have so-called monoallelic cells which express a single HLA allele, and we express the Cas9 in a set of these cell lines, and then look at class 1. So we don't only look at class 1, we look at class 1 after it has been expressed in the way it would be expressed in vivo. And the other issue with Cas9, which is also very interesting and understudied and requires a lot more study, is we study Cas9 with respect to off-target editing. And when we look at off-target editing, we look at it in the context of what that's going to do to the gene. Will it make that some gene more cancerous, or will it hit some gene which is an essential gene, etc. There is another dimension of off-target editing, is that you create neoepitopes, and you're going to probably create, if you do off-target editing in a human, you're probably going to create some kind of, at least there is the possibility that you will create some kind of an autoimmune disease by creating epitopes where they didn't exist before. So, I cannot sit here and prescribe that this is a better method or that is a better method. When you choose a delivery method, you're using it for a host of reasons, which is best for your program. But all I'm saying is that, whatever method you use, like just as you were being a little thoughtful about the different immunological consequences of whatever it is that you do, that is very, very important. You cannot just, pick and choose that, these were my learnings from therapeutic proteins, and I'm blindly going to apply them to novel modalities, and that's not going to work. Not with novel modalities, no.
Thank you. I want to mention, recently we have a lot of other enzymes being developed, identified and developed for the gene cell therapy, not only Cas9, Cas12, and other new enzymes. But we're only thinking about editing efficiency or off-target risk. We're never thinking about the immunogenicity issue. But I think we know the sequence. We definitely look into that in that dimension as well. Thank you.
Yeah. Thank you.